Okay, our last speaker before our open discussion is Paul Have Talk Will Travel Rogers, who is standing in for Jim Catlin, who is sick and wasn't going to be able to present today. Paul has a BS and an MS in Geography from Utah State and University of Wisconsin-Madison, respectively. He has a doctorate from USU in ecology. His primary area of study has been human impacts on vegetation in the western United States. He's worked with the U.S. Forest Service for 16 years, conducting monitoring activities and publishing results from the interior west of the U.S., in Eastern Europe, and in East Africa. His research on lichens in Aspen has taken him around the region and as well as northern Sweden. He is currently an adjunct assistant professor in the Department of Environment and Society and Wildland Resources, and he is the director of the Western Aspen Alliance. Paul.
I think something that's often confused is you're involved in this Western Aspen Alliance, and all you're ever saying, Paul, is Aspen, Aspen, Aspen. We need to be very clear that this isn't a tree, it's a system. And so it's not just an infatuation with a tree, but we're talking about a whole system here. Speaking of systems, I'm going to talk about our research project in Cedar Mountain that we've been doing over the last couple of years, and it's ongoing. But I'm going to try to make this a little more interactive. I'm feeling fairly punchy at this point, and so maybe you are too. Is this a forest? A little feedback here. Is this a forest? Yeah, it's a forest. You think so? It looks a little like a city park to me, maybe a cemetery. I don't know. But are all the forest components there? And so that's not just a rhetorical question. It's something that we want to think about as we go through this. Also, I've been told this talk, having been notified last night that I'd give it, was not exactly defined, exactly put together to address wildlife issues. So I won't say the word wildlife that much. Also in Utah, I've been told I'm not supposed to say the word wolves very much. So if you hear me say wildlife, if you could just give a little howl out there once in a while, then I don't have to talk about wolves, but they'll be a little bit more interactive, and there are all wildlife species somewhere. Also, we are residing right now in one of the northern 6 counties, which is covered by the federal wolf management plan, and we're not in southern Utah where it's not. So to move on here, and I think I figure this out. This project is going to address predominantly forest structure, and then we're going to talk about the part coming up, which is the really interesting part, a landscape analysis of genetic variability. So I'm going to give a little outline and here where we're headed. We're going to talk about why Cedar Mountain and what that is, what the project objectives are, the stand structure assessment, the part 1, and then the part 2, the genetics of a stable aspen community. I also need to define stable aspen community. This is a non-cereal community for those who are not vegetation people. Almost pure aspen without conifers. That helps simplify these things. And there's at least 2 main different types of aspen communities. Actually, we're working on a paper now talking about 8 to 10 different types across western North America. But this acts very differently ecologically than cereal aspen communities. And that's a really important basis of this study. So, first of all, what's the problem? This is Cedar Mountain here. You can see it's a time lapse here. We have a die-off, or it looks like the stand of aspen's dying here, and it's going, gone here. So that's happened in a period of about 12 years. What's going on there? Also, I want to, a little sidebar here, put this in a backdrop of this idea called sudden aspen decline. So we're going to kind of bounce off our results against that terminology. Which I'm not a great advocate of, but we'll see how that fits into this. This has happened here over a 10 to 12 year period. Is that sudden or not? We can talk about that too. So we need to talk about extent and timing in incidents like this. What's going on? What are the mechanisms? Why? So there's certainly a drought element here, probably climate related. Maybe some ungulate herbivory, some non-wild ungulates, and then some wildlife, perhaps. And then we have these incidents of disease and insects, and it's a whole complex of things. It's not easy to just point to one factor in that realm. And then finally, what is this whole bit about genetics and how might that affect things? So, why Cedar Mountain? This is a really cool place and it's very unique, I think, in the West. But just briefly, this is a large plateau just off the south here is the beginning of Zion National Park. This is the upper drainage of the Virgin River. So we have a high plateau, but what really makes it unique is it's more or less the same elevation across this 275 square kilometer area, but it's mostly privately owned. So a whole different set of things going on there. And that's what really makes it unique. So why this area? It's a large accessible aspen landscape. Very cooperative private landowners and local support for trying to figure out what the problems and issues are there. And then it's a unique laboratory of pure, I'll use that term, pure aspen and stable aspen interchangeably, and nearly so. And we're going to first address how nearly. The objectives, a landscape level study, and I'm sort of a real stickler for what's defensible science and what isn't. And a systematic or a random study over a large area is where a lot of my training is in monitoring techniques. And so how do we put that in and make it defensible? We're going to go into that in a minute. But what's the level of purity? We want to determine that right offhand. Because we think we're going to be studying a large landscape that's different than northern Utah, that's different than most of Colorado, Wyoming, and the surrounding states. So we want to define that a little bit. So some really basic traditional stand measurements here. But we want to get at the level of regeneration, and then some visual estimate of tree layers, because that turns out to be very important in pure aspen stands. The number of layers in there is an indicator of health. The second part of this is looking at the genetics, and we want to know about the genetic diversity at the stand level and then across the landscape. Does genetic composition vary among stand layers? So now we're going to talk about stand layers being a surrogate for time. The little guys, the juveniles, or the adolescents and the mature trees. So we'll go into more detail on that too. And then are some clones more susceptible to bark beetles than others? And there's a whole bunch of causal factors, but as we start to tease this apart, it gets quite complex. Maybe not as wicked as Jessica would like, but complex. This is Richard Gardner is in here somewhere using the giant slingshot thing to get samples, sort of shoot a weighted string over the leaves up in the canopy and pull those down for taking to the lab for genetics sampling. And this is the work in progress, so I want to make that clear. Part 1 is pretty much complete. Part 2 is in progress. I originally designed this study for an international presentation, but it's just interesting to note the worldwide distribution of aspen. Predominantly 2 species, Populus tremuloides, Populus tremula. There are some smaller species distributions in Eastern Asia. And then most people know where Utah is, but here's Cedar City right here, Cedar Mountain adjacent to Cedar City to the east and south side. So this is a systematic study. Set up, we take a grid across this vegetative type that was by the Southwest Gap Analysis was typed as aspen. We select all these in the office in a systematic way, and then we sub-select about a third of those randomly so that we've got an evenly distributed sample. 83 plots total. This is the variables we collected just very quickly. 31 plot level variables, 8 tree level variables, and 9 genetic samples. And this is year 1, remember. So, just to give away a little bit of the punchline here. Here's the purity question, number 1A that we're trying to. Which are the stable stands? The larger the circle, the more pure the stand, and that's what this is here. I think about 85% of our stands were only aspen. No other tree species in there. The results here. On stand structure, looking at some of these damage indicators, or the damage to the bowl, there's not really any strong signal there. Crown dieback, this is a typical profile that we see for crown dieback across the country, and then also some of my work overseas, but very little dieback for the most part. We're trying to get at the sudden part, remember. So that's where you see sudden death is a quick dieback, usually outer and upper foliage. And then the dead trees, again, nothing really strong there, so we're trying to get a snapshot in time. What percentage of the trees are dead now? We didn't see any massive die-off. We didn't see sudden aspen decline in this area for the most part. Remember, I'm talking about a whole landscape and a sample. Individual stands might show these signals, but not at the landscape level. So the take-home message from all of these different things, with low foliage dieback, low mortality, and a moderate amount of boll infection, and that's fairly typical. Aspen is easily infected. There's a fairly high rate, somewhere in the range across several western states of 50 to 60 or 40 to 60% of aspen, mature aspen bolls have some sort of infections in them. So a different shot at this is a subjective look at how many layers are in a stand. Not a great signal there. Most have 1 or 2 layers. We'd want to see more than that. Typically in pure aspen stands, a multi-layered profile, but a fair amount had 3 layers. Maybe a more telling signal is the amount of suckers per hectare, and most of our plots, 43%, have what we consider be below a healthy threshold of reproduction. Now this is the suckers, so this is the little small ones. And I want to distinguish here between regeneration and recruitment. Regeneration being the initial suckers coming up, recruitment being those that make it above browse height and start to compose those intermediary layers. So we did a principal components analysis on this. And what we found here, what we're doing is highlighting the plots representing combined measures of healthy regeneration. So we went through all of the variables that we collected, and basically this is the eigenvector loading. It comes down to the only significant variables being related to regeneration and recruitment, so a combination of those things. And what we've done here, because we always like to look on the bright side of life, as Monty Python says, we've highlighted that smaller group of plots, of the 83 plots that are adequately regenerating and have recruitment in them. So we have some obvious clustering here, some statistically significant clustering, and this, if you can't read these here, this combined variable is the basal area of saplings, number of saplings and number of suckers as a combined indicator, you might say, or principal component. So the take-home message there is a low level of regeneration is sort of the overriding message or measure of the condition of this landscape. So clearly we're going to get to the why part at some point. So we see this everywhere. We've seen some pictures of this today, repeated browsing. But is it these non-wild ungulates that are the problem? This area has had long-term, over 100 years of sheep grazing and a lot of the plateau. Or this, well, is this wildlife? Seems that it is. I sometimes wonder at the manipulations that go, how wild is wildlife? But that's a whole other subject. I should back up here and zero in on this. Another exclosure, it's a little hard to see, but here's the fence around the stand, lots of regeneration. The surrounding hillsides, no regeneration. We've seen that message over and over again today. And then let's just go quickly on to objective 2. So we did a systematic measurement across the whole landscape. This is a 1 hectare area. This is where we did all our tree measures. Now we're going to move on to the genetic sampling, which we did at these X's, 9 samples in year 1. And so here's the genetic variability, and the way to read this is the number of plots, and the clones per plot. Most plots have 2 or more clones per plot. So it was often thought that, clones covered large areas, but there's a little bit more variability on landscapes we are finding than we thought. So 78% of plots, multiple clones. I should have acknowledged earlier, but a lot of the work and I'm weighing on here is Karen Mock's work here at USU. She's a geneticist who's done a lot of different work, but particularly some particularly interesting things with Aspen. This is not Cedar Mountain, but this is sort of illustrative of what's going on in other areas. This is actually right up Logan Canyon, but what we see is well, now I lost track. We have different symbols for different what's the word I'm looking for? Different genotypes. And we have I believe, someone's on the audience that can shout it out. The white is the diploidy and black is triploidy. Are you here, Karen? No? Is that correct? So, when we're not doing much and sitting around, we speculate a lot about this pattern. I have some ideas on this, perhaps more pure stands and more serial stands. That's simply a theory. But the point is here, we need to point that there's a lot of variability on the landscape. And that's just what I wanted to show. And that was similar to the results, the initial results, at least on Cedar Mountain. And this is just a landscape level picture of that diversity, number of clones per plot. And so you see that there's a fair amount of diversity, maybe less so here, so maybe we're seeing some geographic or biogeographical pattern there. We don't know yet. So this is just showing that there is diversity on the landscape, sort of part of the first question. But the other two questions, we're starting to look at vertical diversity, so it's kind of really cool to think about how these might change over time. And again, looking at the small suckers, the medium size, the medium layer, and the upper layer. And I just wanted to show folks sort of where those sample plots are on the landscape. Again, randomly chosen there across the landscape as best we can. And then doing this bark beetle surveys on fewer plots. And I don't know if you can see those green circles, but I think we have about 8 or 9 of those plots across there. So this is sort of still being analyzed, this data. And I just want to sort of discuss this a minute further. So we're taking leaf samples, we take them to the lab, get their genetic makeup, and we do a small, medium, and large tree at each point. And now we've intensified this grid. This is still 1 hectare, but now we have 20 sample points. That was 25, I think that's wrong. At each point. So we're getting more intensive at the places we've gone because we really want to discern what's going on here. It'd be really interesting to know if this genetic diversity that's happening down here in the sucker layer is preserved through time. If not, why not? And that gets at some of these ungulate questions, perhaps even wildlife questions, about what's the browse preferences here, or not, or perhaps there's some other agent going on there. But we believe that there's some genetic and phytochemical link to that. So to be determined here. And then this is the other part of the final question for the genetics is also the leaf sample and looking at these aspen bark beetles. So a tree without aspen bark beetles, tree with, and then is there any difference there? Are there any correlations in terms of the genetic makeup? And that could tell. So maybe there's some preference feeding there by these tinier herbivores. So just to recap there, we feel like we've answered that question. Looking at the vertical diversity, causal agents, and the susceptibility to these different agents. What we really want to get to at some point, just need to think about is this mortality, agent, genetics, and climate connections. Or are there any? But perhaps we could go down that avenue at some point. But again, we're still in the works on that one. I just wanted to, I don't have the handouts, that's incorrect there, but these papers are out that are, the one I showed you with the map and Karen's work is very interesting and I think sort of a page turner in terms of changing our minds about some of these perceptions. And then this paper is already published on the stand structure, which was the body of this talk. So, a lot of people to thank here. I think I'll just let you read through those. Several of these people are in the audience, but a lot of help here from many people. This is a big project. And then I will take any questions. Just another, this is not an exposure, but you see some clear differences to sort of an incidental fence here with our last visit about a month and a half ago to Cedar Mountain. So is this genetic makeup? Is this fence? You decide. So I'll take any questions.
Can you tell us a little bit about fire history on Cedar Mountain?
excellent question. So again, typically, and this isn't across the board, but the pure and stable stand, I talked about them having a different ecology. And a lot of times, fire plays very little of a role in that, or the very rare events. Because somebody termed these forests at one point asbestos forests. They're very difficult to burn, but at certain situations they will burn. And we've had a lot of experiments, particularly around central and southern Utah, with trying to burn them and just working to burn these forests and not having much luck. But with the right drying conditions in late summer, early fall, you can get them to burn. But in this case, anecdotally, there hasn't been any evidence of fire at all on the plateau. We don't have data to support that. But in terms of the initial soil samples, no charcoal, no nothing.
So you have no estimate of a fire interval?
That's correct. So one of the take-home messages and the motivation of this alliance is, let's get off of this path that all Aspen forests are the same. And this does not have the same solution, perhaps, that you might have for a zero condition where, maybe fire suppression had a role, maybe it didn't. But in this case, probably very little role. Yes?
So you're finding greater regeneration in the stands with greater genotypes?
That she asked if they're finding greater regeneration in stands with more genetic variability. No, I didn't say that. We don't know that yet. What we're trying to do, maybe I didn't state the question clearly, what we're trying to do is determine what that genetic variability is at different levels. And those different levels are surrogate for time. So we don't have an answer to that, but I didn't mean to imply that. Yes?
You said that 43% of your plots is below a healthy threshold level with records for that there.
Yes.
What's the definition? How do you define your threshold?
He asked, what's the definition for that threshold for, some people use sustainable or self-replacing, but basically that came from the literature, is the short answer. Some of the work that Walt Mugler did and others, and now it seems to be used fairly consistently in the literature, but that is certainly something that's up for debate, and maybe that's what you're hinting at. But that's the best we have to go on at this point. That translates into 500 suckers per acre, just in case you're wondering. And I'm advocating a scale of sort of at-risk, not self-replacing and self-replacing. And those, I could talk about those cutoff numbers with you at some point. But that comes from the literature. It's the short answer. Yes?
Okay, in the areas that you're getting the generation, are you getting any from seed or
excellent question. So she asked if the regeneration we're getting is any of it from seed. And this is another important finding that I sort of skipped over fairly quickly, is that there is perhaps more regeneration from seed in having more genetic variability. We're not ready to answer that question now. We don't know that. I think we'll have a hint at that once we get through that analysis of the genetic data. But I don't have an answer right now, but that's a very important question for us to keep in mind. More?
So one more problem. What was, maybe I missed this, but what was the reason for the die-off in those first pictures that you showed us? 1990, 2002?
That's what we're trying to answer.
You're still working on it.
That's right. And there's some related work going on here, but our best guess at this point, not from this study, but from some other work, is sort of drought and signals. But, and hopefully we'll talk about this a little bit more tomorrow, is whenever we have these natural disturbances in droughts or whatever, a lot of times it's how we as humans have set up these systems. Are we managing for sustainability or resilience, resistance or resilience? And a lot of times we set things up at the edge, and then the disturbances tend to push them over the edge. That's my feeling. So, but that's another work and another talk. But the short answer is probably a longer term 20 to 30 year signal of drought is pushing that die-off in those particular forests. And that's some work I should mention of Ron Ryle and his graduate students. Oh, one more question.
One more. Did you know when you collect the data, did you know if the recruitment or all the metrics you took, if they were in an opening or the interior of the snake?
He asked if the recruitment, and I'm not sure if you mean that literally or you mean all regeneration, was it openings or interior with stand? We tried to locate all sample plots within stands. We didn't have any large openings, but some stands are more open than others. So I'm going to dance around an imperfect answer that I don't really have for that, and because then we have to start defining what an opening is, how big and all that. So I don't really have an answer. Maybe you're suggesting there's some sort of gap replacement or larger openings. I don't know. Colonizing of meadows, but we didn't measure that as a variable specifically.
Thank you. Thanks, Paul.